But Brian is very well recognized for many, many different works in biophotonics. And he's been working for decades in photodynamic therapy, many aspects of it, and also some photodiagnosis. In the early stages, he did a lot of that and still doing many work on that. He is a physicist, originally from Scotland. And in the early 80s, he started a program in translational research and collaborative clinical trials for photodynamics, especially for brain surgery, prostate, gastrointestinal cancer. And he has driven and developed many, many tools for fluorescence guide spectroscopy. And more recently developed optical imaging to guide surgery that's being evaluated in clinical trials for head and neck, and also brain surgeries. Nowadays, he has expanded to include a nanotechnology application in cancer treatment, as well as diagnostic. He is currently head of the Division of Biophysics and Bioimaging at the Ontario Cancer Institute and professor of medical biophysics in the Faculty of Medicine at the University of Toronto. Brian is being a mentor for many, many, many groups around the world, including our group. I still remember in the SPIE meeting when we first met, and Brian was very nice. And since then, he became a friend of many Brazilian groups here in Brazil. And it is with great pleasure that we have him to give us this plenary talk on the SBFoton meeting here in Brazil. So Brian, thank you very much, and you have the time to go.
Hey, Vanderlei, thank you so much. You can hear me okay?
I can hear you perfectly.
All right, so thanks. I'm so sorry not to be able to come to Brazil in person to attend the meeting, but this is the next best thing. So I thought I'd talk about two different aspects of one common problem in the use of optical techniques for cancer treatment. And since I wasn't sure what the audience would be, let me just first of all say a few general words about the advantages of photomedicine, why use light in medicine. And I've just broken this down in the following way, that if you look at the energy of visible and near infrared light, then it's very important that that corresponds to molecular transitions. And so you're able to probe cells and tissues at the molecular level for doing analytic and diagnostic techniques, or you can alter molecular function for treatment. Again, in visible ranges of the order of cell size, which allows high resolution imaging for analytics and diagnostics. And of course, the whole of optical microscopy is very important for that. A very nice aspect of light in interacting with cells and tissues is that there are many different interactions: absorption, elastic, inelastic scattering, polarization, interference, nonlinear effects. And so that means that in practice, there's a lot of biological information that you can access. It's non-ionizing, therefore it's safe for human use, relatively low cost and compact technologies. And in practice, that means that these technologies can be disseminated and adopted into clinical practice or adopted for research purposes. And also, in most cases, the optical techniques are compatible with other biomedical technologies. So you can use optical techniques at the same time as you're using, say, magnetic resonance imaging, or at the same time as you're doing surgery or you're doing drug treatment. And so that allows you to integrate and develop hybrid approaches to solve problems. The fundamental limitation of using light in medicine or a fundamental limitation, comes from the fact that light does not penetrate easily in tissue. And so if we imagine light hitting tissue, there are many processes, as I've just described, but in terms of the ability to penetrate into tissue, it's the absorption and the elastic scattering that dominate that process. And if you plot the if you like, the intensity of light on a logarithmic scale as it penetrates into tissue, you find that it's essentially exponential. The so-called effective penetration depth depends on the absorption coefficient and the so-called transport scattering coefficient of the tissue, which is a scattering after accounting for the directionality of scatter. And that's just a few millimeters in tissue in the visible and near infrared. The absorption comes from multiple components such as hemoglobin, water, melanin. The scattering comes from refractive index mismatch at the microscopic scale. But it means in practice that light is diffused in tissue in a few millimeters. And so from a diagnostic or imaging point of view, you lose spatial resolution and contrast. And from a therapeutic point of view, it limits the effective depth that you can reach or the volume of tumor that you can treat. And so I want to consider two different ideas that we've been working on to overcome that limitation for a couple of different applications. And the first is in the treatment of melanoma using photodynamic therapy. And then the second is this idea, an emerging idea of using radiodynamic therapy, which means combining some of the advantages of light activated treatment, but using high energy X-rays that can penetrate deeply. Just to remind you, I know there's been a talk on this already, but if you look at the basic photophysics of photodynamic therapy, you have the ground state of the photosensitizer molecule absorbs a photon of light, is raised to an excited singlet state. You can re-emit the energy as fluorescence, and that's what you see here. For example, this is a tumor undergoing resection. This is in the bladder, and you see very beautifully the in this case fluorescence allows you to visualize the tumor clearly. And that, as Vanderlee said in the introduction, is quite a large area of our research, but I won't talk about that today. The singlet state can undergo intersystem crossing, in other words, a spin flip, to an excited triplet state. The reversion to the ground state is quantum mechanically forbidden, and therefore this is a very low probability process. It's slow, and so this triplet state lasts for long enough to interact either directly with biomolecules or to interact with ground state triplet oxygen, producing radical species that can then destroy cells and tissues. And so the idea here is that we give this patient the photosensitizer, we wait some time for uptake into the target tumor, and then we deliver light, usually nowadays by optical fiber or by LEDs, to activate through this process. So the first way in which we're looking at overcoming the light penetration here relates to melanoma. This is a collaboration between Canada and Brazil. That's how it started, with collaboration with the group in San Carlos, but now also involves people in the UK and in Ireland. So this just lists the people here. So we have our people in Toronto, including Leila Pires, who is from San Carlos and is a very important person in our lab. It includes Vanderlee and Christina Sebastião and others in San Carlos. Also Anna Gabriela Salvia at Brazilian dermatology, and people in France and the UK and Ireland. So it's quite a nice international collaboration. So first of all, we start with melanoma in the skin. This just shows an example of melanoma. I think everyone is familiar with this. This causes about 55,000 deaths worldwide. And what we really need here is a minimally invasive treatment that destroys the full thickness of the tumor and reduces tumor progression and spread as an alternative or adjuvant to surgery. The other type of melanoma I'll talk about is ocular melanoma, much less common, but nevertheless a difficult disease. This is a tumor in the eye. And what we need here is, again, a minimally invasive treatment, but we want one that preserves sight, because often the eye is removed as a way to treat this disease. But we also want to reduce the possibility of spread outside the eye and progression. So let's start with cutaneous. As you may know, and certainly the people in Brazil know, that the group at San Carlos has done a spectacular job in disseminating PDT for basal carcinoma. This is non-melanoma skin cancer, a very common disease, very difficult disease. And through the development of fluorescence and LED treatment light sources, there are now many centers have been set up in Brazil, and this is now disseminating up through other countries in Latin America, and this is a real success story for PDT. However, when you try to do this in melanoma, it works very well. There's poor efficacy. You can get local regression, but not complete response. There are a number of reasons for that, both physics related to being the penetration problem I just spoke about, particularly because of melanin. There are also some biochemical and biological effects. So our primary idea was that we could overcome this limit of the melanin by so-called optical clearing and possibly whitening. And what does optical clearing mean is illustrated here, where a tissue is exposed to or soaked in a medium that has a refractive index, which is comparable to the refractive index of tissue structures, which is around 1.4. And by doing this, you are able to reduce the refractive index mismatch and essentially virtually eliminate the scattering or markedly reduce the scattering. And if you go back to that earlier formula I showed you with the square root of 3 times mu A times mu S prime, that means that you're effectively increasing the penetration depth, even if you do not change the absorption. It's possible to change the absorption also by removing or fragmenting the pigments, but I won't talk about that today. So we'll just talk about this optical clearing idea. And just shows this just in a melanoma model. In mice, this is optical coherence tomography, showing you get increased penetration. You can get increased, if you can compare here with here, you see that you get much clearer, higher resolution imaging at depth, which indicates that there's much less light attenuation. So study, and for this we used 2 different photosensitizers. One was targeted to the blood vessels of the tumor, and the other is targeted to the actual tumor cells. So we had these tumors growing in mice. We either gave no treatment or we gave PDT with the tumor targeted, or PDT with tumor targeted after clearing, and then the same with the vascular targeted. And then finally, we tried the combination of both tumor targeting and vascular targeting. with and without the optical clearing. And we looked at a number of endpoints such as tumor volume and histopathology. So I wouldn't show you all the results, but just an example to show you the volume of tumor. So here we follow the animals after treatment and measure the tumor volume. And this is the tumor volume. So these curves are for melanoma does not have melanin. So this was a control. This is so-called non-pigmented melanoma. And here you see how the tumor grows in the absence of any treatment. And here is the tumor growth after treatment with the vascular targeted agent with and without the optical clearing. And the same here, this is with the tumor targeted agent, and here is with the combination agent. So what you see from this is that in the absence of melanin, this is actually pretty effective. So the tumor growth is essentially completely eliminated, and it really doesn't make much difference whether or not you use the optical clearing. So this is a bit like the basal cell carcinoma that I showed you that's now being used routinely. If you don't have a lot of melanin pigmentation, then you can treat these tumors successfully. However, when we go to the more standard melanin-containing tumors, again, here we have the growth curve. These are very, very aggressive tumors. They grow very quickly. This is what happens when you use the vascular targeted agent with and without the optical clearing. So if you do not use the optical clearing, you get a little effect on the growth. But if you use the optical clearing, you essentially can control the tumor. And the same with the tumor targeted agent. If you don't give any treatment, the tumor grows. If you give the tumor targeted agent by itself, the tumor still grows. But if you give optical clearing plus this treatment, then you can essentially control the tumor. And the best result is obtained, in fact, if you do the combined treatment with the two different agents plus optical clearing. So this is a very, very encouraging result. And we had a nice paper published last year in Cancers, which was a collaboration between the group in San Carlos and our group in Toronto. In parallel with this, we started some collaborations with the group in Brazil to do the first ever human studies of optical clearing. So this, in fact, this is again OCT. This was actually the first patient ever to have optical clearing. Although optical clearing has been around in the literature for many years, it has never actually been used in patients. So this was the very first patient in Brazil who had optical clearing. What this has shown is that we probably need to do a little better because when we give the optical clearing agent, it goes onto the tumor surface and therefore is limited. And so we're working with a group in Ireland who have these microneedle devices. These are microneedles made of a biogel that contain the sensitizer and the clearing agent. And this is pushed onto the tumor. Each of these tiny little needles penetrates into the tissue and is able to deliver both the photosensitizer and the optical clearing agent to greater depth. So that's the skin story. Let me talk about the eye story. And the eye, here's a tumor. This is really pretty difficult to treat. You have to get very precise delivery of the treatment to this area of tumor without damaging the rest of the very sensitive structures of the eye. And so for this, we decided to use 2-photon PDT. To remind you, under normal PDT, you have a single photon of sufficient energy absorbed to excite the singlet state. And if you look at this from a light distribution point of view, for example, in confocal microscopy, if you use 1-photon confocal microscopy, you get a beam of light, although it's focused, you get activation above and below the focal plane, whereas with 2-photon, because this is a nonlinear process, therefore you need to have very high instantaneous intensity through using something like 100 femtosecond laser beam. You can get exquisite diffraction limited control of the light delivery. And so we have that. This is a 2-photon confocal microscope. This is the objective lens. This is a mouse model with melanoma growing in the eye. And we basically are able to put a point of treatment into the eye and scan that through the tumor volume. These are histopathology slides. This large mass here is tumor. So this is untreated. And then at 48 hours after treatment, if you give light only, there's still lots of tumor here. If you give a low dose of PDT, you get some tumor response. And without going into the details, if you give a high enough dose, in this case of the vascular targeted, then you can get complete destruction of the tumor, and you see that the rest of the eye is preserved. So that's very nice, but it raises a question, or a couple of questions. First of all, is that nice result obtained because the intrinsic cytotoxicity of 2-photon PDT is the same as 1-photon? And is that intrinsic cytotoxicity, the ability to kill the cells, is that different in pigmented and non-pigmented melanoma cells? After you've accounted for the effects of the melanin absorption and this effect on the penetration depth. So if you look at this from the photophysics chart point of view, we're talking about doing 2-photon activation. So we reach the same excited singlet state. And so from this point onwards, it should be the same. So it shouldn't make any difference whether or not you do 1-photon or 2-photon, as long as you've taken account of the penetration depth. The photophysics should be the same, the photochemistry should be the same, and the outcome should be the same. So here is the study looking at that. So this is in cells. So this is a layer of cells in a dish. And this just shows you the treatment with 1-photon, treatment with 2-photon. If we look at the cell survival curves here, so the y-axis is cell survival. So 100% means you haven't killed any cells. And the x-axis is the number of scans. This is again done under the confocal microscope. So this is equivalent to the light dose. And you find that, and there's two different cell lines here. One of them is pigmented and one of them is not pigmented. They are otherwise identical. And you find, in fact, that the 2-photon, if you do light only, there is a little cell kill because you get, these are very intense pulses of light. So there's some photomechanical cell kill, but it's only about 10 or 20%. And there's no difference between the cells that have got melanoma and the cells that don't have melanoma. On the other hand, if you look at 2-photon PDT, you find this very surprising result, namely that if you have melanoma in the cells, then it's actually much easier to kill them with 2-photon than it is if they do not have melanoma. Again, we've accounted for light penetration, so we don't need to worry about that. So that was a very surprising and counterintuitive result, that you would get 2-photon is more effective at killing the cells than 1-photon. And so we have been puzzling, scratching our heads as to why that is. So there's been lots of conversations between Toronto and San Carlos to try to work out what's happening here. There are a number of possible reasons. The pulsed light, either photomechanically or photodynamically, can rupture the little organelles in which the melanoma is held. and that is known to release chemicals that can be cytotoxic. So this might be an additive or synergistic cell kill. PDT under 2-photon illumination can actually generate single oxygen from the melanin itself. And then the third possibility is that melanin under 2-photon or UV activation actually fluoresces. And so it gives off fluorescent light, which itself could be reabsorbed by the sensitizer and add to the cell kill. So we've been trying to work this out. These experiments are actually quite complicated to work out what's happening. So that's a work in progress. Some recent very interesting evidence that this is photophysical and photochemical comes from a comparison that we just did with our standard photosensitizer. Another sensitizer that was made for us by collaborators at Oxford University. It's a dimer, and it's designed to have very, very high 2-photon cross-section. Most photosensitizers have relatively low 2-photon cross-section, but this was designed to have very high. And very interestingly, it turns out this is just a plot you've just seen, where the 2-photon is much more effective than 1-photon. If you use this sensitizer, there's actually there's not very much difference. So in other words, if you use a sensitizer where the 2-photon cross-section is high enough, then that seems to swamp the melanin-mediated cell death. And it doesn't matter whether or not the melanin is present or absent. So that's some clue as to what's going on here. Anyway, this is very much a work in progress. So the second topic is also an international collaboration between Canada and Australia and the US, with our group again, a group at University of New South Wales in Sydney, Australia, and then a group at Dartmouth College in the US. And again, it relates to this question about the limited light penetration, but now not just uniquely for melanoma, but for any tumor. And just to reiterate, it's challenging to treat tumors with PDT that are deep-seated, large volume, or disseminated. It's not impossible, because you can use optical fiber delivery, and that's important to emphasize, but it is challenging. And so the concept of radiodynamic therapy is one that emerged a number of years ago, which is we would, rather than using an external light source, like here, like a laser or an LED, we would use ionizing radiation, either in the form of X-rays, high-energy X-rays, or in the form of radionuclides. The advantage of that is you get deep tissue penetration. As you may be familiar from radiotherapy, these are radiotherapy beams in tissue, so you can penetrate fully into the body. But by using these X-rays, not themselves to kill the cells, but to activate the photosensitizer, we hope to retain some of the selectivity and the biological effects, the biological advantages of PDT. There's actually 3 different ways in which this can work. The first is direct activation of the sensitizer by the X-rays, or more correctly, by the second electrons generated by the X-rays. The second mechanism is Cherenkov radiation. You'll be familiar with Cherenkov that you see as the blue glow in nuclear reactor. But in fact, if you take standard high energy X-rays used in radiotherapy, then that actually generates Cherenkov light in tissue. And that's being used, was developed by Brian Poga as a way of imaging the radiation beam as it hits the patient. This is a breast patient being treated, and this is actually imaging of the Cherenkov light generated in the tissue. And the third possibility is to use a material that is a scintillator, absorbs X-rays and gives off visible light. And of course, there are many, many scintillators So that's the third possibility. We just recently written a very large review paper on the mechanism for how to do this. So here's some examples. This is, again, is very much a work in progress. It's much less fully developed than the melanoma story. But here we've used nanoparticles with verteporfin, that same photosensitizer as we used for the melanoma studies. In this case, the nanoparticles are targeted to the tumor using an antibody. This just shows a singlet oxygen now as a function of X-ray dose rather than as a function of light dose. Just to give you a sort of calibration here, a standard dose of X-rays for radiotherapy is that a dose of 2 gray, typically around 2 gray, are given every day for about 30 days. So what we're interested here is being able to use just less than, say, 10 gray of radiation dose to get an effect. And here is some cell survival curves showing that with the black curve is the X-ray response. You see these tumors, the pancreatic tumors, are not very responsive to X-rays, but when you add the nanoparticles themselves, have no effect. When you add the sensitizer, you see this extra cell kill over and above what you get from the X-rays alone. This is an example from our own lab. This is a linear accelerator, a clinical linear accelerator in this phantom. So this is a translucent material. So there's a lot of Cherenkov light that's generated in this phantom. The cells are inside here, and here you see the survival curves. So you see there's lots of cell killing going on, or some cell killing. One warning on this for those of you who know photobiology is that there may be a contribution here to the extra cell kill from the fact that X-rays themselves can cause permeabilization, in other words, leakiness of the cell membrane, which may allow you to get more of the sensitizer into the cells. So there's a little artifact that needs to be sorted out there. Here's a nice example from literature from Sam Ashalefu's group on using Cherenkov with the radionuclide. So in this case, they use a positron emission radionuclide and nanoparticles as the photosensitizer. And again, here's the in vitro, the cellular toxicity with increasing dose of radionuclide. And here's in the tumor model, the growth curve without treatment, and then with the radiodynamic therapy treatment and the corresponding extension of survival. So that looks very nice. The problem is that for this to work, it needed the injection of the radionuclide and the sensitizer into the tumor itself in order to get a high enough effectiveness, and that's not a very practical way to do it. I'll come back to that point. Here's an example of scintillation. This is work that we did with the group in New South Wales. So these are cerium fluoride nanocrystals, about 5 to 10 nanometers in size. Cerium fluoride is a nice X-ray luminescent material. So you get, in fact, UV light. The light, the wavelength's a bit short here. Actually, we'd prefer it longer, but there's UV light emission from the luminescence. So that light is given off, is absorbed by the photosensitizer, and then that generates the singlet oxygen. So here you see the singlet oxygen generated and this corresponding cell kill. And there's some nice physics you can do here to work out how much of the energy is actually utilized in this process. Here's an example of proton activation. So this is a group in Scandinavia who used a proton beam to actually activate the photosensitizer. And again, here's a nice cell survival curves in brain tumor cells. If you look at the energetics of this, you realize there's an issue. If you do radiation therapy, a total dose of radiation therapy is about 100 gray. I told you it's 2 grays per day for about 30 days. So, 60 gray, 80 gray, 100 gray. That's about 0.1 joules per gram of tissue. It's about 10 to the 12 absorbed X-ray photons per gram. If you look at light-based therapies, any phototherapy, whether or not you're talking about photodynamic or photothermal, photoablation, you need about 1,000 times as much energy absorbed, about 100 joules per gram typically, or about 10 to the 21 absorbed visible light photons per gram. So there's something happening, there's something different here about giving ionizing radiation versus giving optical radiation treatment. There's some energy gap issue. And the real reason why this is so much more effective on an energy basis than phototherapy is, in fact, that radiotherapy causes DNA damage, whereas phototherapies generally do not cause DNA damage, they cause membrane damage or protein damage. And that's much less efficient way of killing cells than DNA damage, because when you do a little DNA damage, the cell is, if it's unable to repair, is unable to divide and replicate, and therefore the tumor stops proliferating. And so there's this biological amplification effect that you get if you target DNA. So we had the idea a few years ago, if this is the case, why don't we target the DNA with the photodynamic therapy, in fact, with the radiodynamic therapy? And that led us to ask this question: can you actually get this to work with a low dose of X-rays? And it's interesting that if you look at the literature, different components in cells, the cell membrane, and so on, can be targeted with PDT, and the different organelles in cells are very in their sensitivity. So here's the work that we did a number of years ago, looking at mitochondrial damage. So here is surviving cells versus singlet oxygen produced by PDT, and you find if the sensitizer is in the mitochondria, it's much more sensitive than if it's in the cell membrane. And there was work done, including work in our own lab about 20 years ago, showing that if you targeted to the nucleus, then you could get a huge increase in the effectiveness of the cell kill. So there's biological amplification from DNA damage if you do phototherapy targeted to the nucleus. And so what we've been doing is using polymeric nanoparticles, and we've added to them an agent to, in fact, a peptide that will target them to the nucleus. So this just shows there was a lot of chemistry to make these materials. This shows this is a stain for double-strand breaks in the nucleus of DNA. And here you see with this particular agent and with radiodynamic therapy, this is actually a 4 gray, so quite a low dose of X-rays, you get this big increase in the DNA double-strand breaks. And if you look at the cell survival, in this case, so-called clonogenic assay, where you're measuring the ability of the tumor cells to replicate, you get this very significant effect on cell survival. And this is with no scintillation and with minimum Cherenkov. So when we add scintillation or we add Cherenkov, we expect that this effect to be much greater still. So we're very encouraged by this, and we have work in progress to try to make, for example, scintillation containing nanoparticles. So these are polymeric nanoparticles that contain the photosensitizer, but also contain the cerium fluoride nanocrystals. And this is our latest results from that in pancreas cancer. You see the cell survival, and in breast cancer. This is using direct plus Cherenkov. So the next steps in this project are there's a lot of optimization still to be done to try to get the maximum tumor cell uptake, the maximum targeting to the nucleus. And then we'll go on to testing in vivo in an animal model for efficacy, safety, and application to disseminated disease. And then we have looked at design for clinical translation, either as a standalone treatment or to enhance conventional radiation therapy. So I hope that these two examples have given you some sense of the fact that even although the penetration of light in tissue is limited, primarily actually limited by the scattering as well as by the absorption, there are ways around this or over it or through this limitation for particular applications. So with that, I'd like to say thank you very much and obrigado.
Vanderlei, can you see the questions?
No. Could you perhaps read them to me?
In the Q&A box.
Okay, so let me see if I can find that. Q&A box. Here we are. So the first question is, what's the spatial resolution for 2-photon PDT, and what's the absorption of the second harmonic for the melanoma? So the spatial resolution for the 2-photon PDT is actually diffraction limited. So that's in practice for the at depth in tissue, the spot size is probably of the order of a micron. Probably, yeah, it's probably not better than micron. I'm not quite sure what you mean by the absorption of the second harmonic for melanoma. Maybe you could expand on that question, please. And then while I'm waiting for that, I'll look at the second question. Comment on the optical tissue clearing limitations, materials and injection. What's the increase in penetration depth? Comment on the use of optical clearing in photothermal therapy. Okay, the optical clearing should be equally effective for photothermal. The tissue limitation, clearing limitations. The materials are non-toxic. So it's a mixture of polyethylene glycol and other agents. People have used different materials, but they're non-toxic and they're transient. They're cleared. Probably if you want to treat very deeply, for instance, as in photothermal therapy, you may have to inject the optical clearing agent. And that should be fine. So we haven't done that, but in principle, you could clear quite large volumes of tissue by localized injection of clearing agent. And the increase in penetration depth, we get about twice the penetration depth with the optical clearing. The Cherenkov light, can the Cherenkov light cause any collateral damage? No. Unfortunately, well, fortunately, the Cherenkov light cannot cause any collateral damage because it's so weak. So, for example, if you look at the case that I showed you of imaging the radiotherapy beam with the Cherenkov in the breast patient, in order to do that, you need to synchronize a very high sensitivity camera with the pulsed X-rays in order to get enough signal. So the Cherenkov light is for radiation doses that are typical of that we'll be looking at. They're of the order of microwatts, and so the damage should be minimal.